Hello everybody. In this lecture, in the next half an hour or so, we will study the basic concepts of how we preserve and process fruits and vegetables by addition of heat. So, preservation and processing of fruits and vegetables by addition of heat is the topic of today's lecture. The concepts which will be covered include principles and methods of thermal processing, blanching, pasteurization, sterilization, aseptic processing, etc. Also, we will discuss important aspects of the thermal process calculations like D value, Z value, etc., lethal rate, and the quality changes during thermal processing, which is a very important aspect. Okay. See, the purpose of the addition of heat or thermal processing is obvious, that it reduces or destroys the microbial activity, reduces or destroys the undesirable enzyme activity, and it causes desirable physical and chemical changes, that is gelatinization, denaturation, etc. in the product to give the desired characteristics. So, thermal processing is divided into two major categories. That is one traditionally used technology is thermal processing, in container sterilization, which is called canning, okay, and the aseptic sterilization, that is the processing, aseptic processing and packaging. And the principle involved in both the processes remains almost the same. These heat processes can also be categorized on the basis of their severeness. They like mild processes include blanching and pasteurization, severe processes include canning, baking, roasting and frying. Okay. Blanching is used for destroying enzyme activity in fruits and vegetables. It is used as a pre-treatment to freezing, drying and canning. Air, water or steam are used to heat the product to about 88 to 99 degrees Celsius, and when the water or steam is used to blanch sample, it is referred to as wet blanching. So, it is a very important operation to improve or to get the desired characteristics in the product after the drying or even after during the canning, etc., or even freezing, that is like freezing vegetables, etc. If it is not this inactivated enzymes are not inactivated, then during the frozen storage it may create some problem. Hot water blanching, in this method the cleaned food is subjected to hot water until the enzymes are inactivated. Okay. That generally pot blanchers are used at home scale. So, it is low capital cost and it gives better energy efficiency. However, there are certain disadvantages like maybe loss of water soluble constituents. There may be risk of contamination if water is contaminated or higher effluent disposal. In this table, I have given that generally recommended blanching times in minutes for the commonly fruits or vegetables like broccoli can be blanched for 3 to 5 minutes. It is blanched or steamed, or carrots like depending upon its sizes, etc., from 2 to 5 minutes. Onion, that is blanched until center is heated like 3 to 7 minutes, or even rings of the onion is blanched just for 10 to 15 seconds. Okay. Steam blanching, that is in this steam blanching the food product is directly exposed to steam to avoid the loss of the food soluble solids. Okay. On commercial level, generally tunnel steam blanchers with product conveyors are used. Okay. CIA Bhopal has claimed developing a steam blancher, that is which is having 100 kg capacity per hour, and it is used for blanching of the cabbage, cauliflower, pea and okra, and they claim that the product color, that is there is a better retention of the color in the dried product. Okay. Pasteurization, it is a mild heat treatment for relatively brief duration to kill the part of the microorganism and to eliminate human pathogens present in the food. Pasteurization may be either low temperature long time, popularly known as LTLT, or high temperature short time method STST, or ultra high temperature method UST. Like in the LTLT method, that is generally it is used for 63 to 65 degrees Celsius for 30 minutes or 75 degrees Celsius for 8 to 10 minutes. That is the minimal heat treatment for grape juice. It is normally done 76.7 degrees Celsius for 30 minutes. The STST process, it means generally 71.7 degrees Celsius for about 15 seconds or so. Grape wines, they are pasteurized at 81 to 85 degrees Celsius for about 1 minute. In the UST, it is 85 to 90 degrees Celsius or more temperature and time in order of seconds. Typical combinations of 88 degrees Celsius for 1 minute, 100 degrees Celsius for 12 seconds or 121 degrees Celsius for 2 seconds, etc. are used. And this obviously the UST, ultra high temperature, provides the best product with better flavor and vitamin reduction, but it is generally expensive process. Sterilization, it is the complete destruction or elimination of all viable organisms in or on a food product. Okay. It destroys yeasts, molds, vegetative bacteria and spore formers and allows the product to store and distribute at ambient temperature with extended shelf life. So, sterilization, there are, as I told you earlier, there are two methods. Traditionally used sterilization process is canning. In the canning, what is done? The material in the prepared form is placed, that is the placing the fruits or vegetable in a suitable container, maybe glass bottle, glass container or metal cans, etc., evacuating and sealing the container, and this is after the container is filled and packaged and sealed, then it is subjected to a desired heat process, that is the heated for desired at desired temperature for desired required time to get the required microbial inactivation. So, in this I have given you a typical fruit canning process where obvious treatments like that is peeling, washing, cleaning, peeling, coring, cutting, slicing, etc., and suitable treatment for the, in the last class in the dehydration we studied that what are the different pre-treatments. So, here also blanching is a suitable blanching method is given, and then finally it is put into the sealed into the can, suitable can, and exposed in the different method. Or similarly a vegetable canning, it is also the process almost the same steps. In the case of vegetables, normally steam blanching or hot water blanching is done, and inside the can, in order to facilitate the better heat transfer, the can is inside the vacuum. So, to facilitate the heat transfer, in the case of vegetable generally brine solution is added. In the case of fruit, generally sugar syrup of a various concentration depending upon the type of the fruits and its acidity is used. Okay. Then aseptic processing. In aseptic processing, the product is subjected to a thermal process to inactivate vegetative cells or spores prior to being placed in the sterile container. That is, here what is done? The packaging material, okay, it is sterilized separately, and the food which is used, the pumpable food, the mostly liquid foods, etc., they are sterilized separately, and then the product is aseptically packaged in the using FFS machine, form, fill and seal machine, okay, where the packaging material comes and then it is formed, packet is formed, it is filled with the material and then finally sealed. So, there are normally for the sterilization of the juices, etc., different types of heat exchange equipments like scrubbed surface exchangers, plate heat exchangers, tubular heat exchangers or steam injection based heat exchangers are used. And in fact, of course, on aseptic processing itself we will take up a separate lecture, and there we will discuss in detail the technology, but here let me tell you at this time that is most of the packets which you see in the market with various brands of vegetable juices, fruit juices, etc., majority of them are produced using this aseptic processing and packaging technologies. Now, let us come to the important aspects of this thermal processing, that is the thermal process calculations. Okay. And for thermal process design, the foods are normally classified into three groups on the basis of their acidity, that is pH, like high acid food, and low acid food. And this, since the pH of a food is an important factor affecting the growth and multiplication of microorganism, so this pH, that is S, it will tell that which type of microorganism will grow. For example, in the low acid food, all the spoilage and pathogenic microorganism can grow, but in high acid food, that is the fruit or which has pH that is more than pH less than 3.7, okay, they will not support the growth of spore forming bacteria. Okay. In acid foods which has pH between 3.7 to 4.7, 3.7 to 4.5, yeast and molds can easily grow in that. So, accordingly, if you know the pH of the commodity, you can sort list, you can find out that yes, which one is the group of the bacteria or yeast or mold which can grow into the commodity, and that accordingly you can process your product and decide the thermal process conditions. That is pH 4.5, which is the dividing line between the acid and low acid food, is slightly higher than the pH that is required by Clostridium botulinum for its growth. Clostridium botulinum is the most heat resistant obligate anaerobe spore forming pathogen which can grow in the low acid food or canned foods. They are normally particularly vegetables which are used for canning. So, destruction of Clostridium botulinum is used as the criteria for successful heat processing of low acid foods or moist. Most heat resistant strains of Clostridium botulinum are type A and type B. This Clostridium botulinum produces a toxin that is which is extremely potent, but it can be destroyed by exposing the toxin to a moist heat for about 5 to 6 minutes at 80 to 85 degrees Celsius. So, in this slide I have given you the optimum pH values for the thermal processing of vegetables, and it decides that is most of this accordingly. So, one can find out whether it is in the category. Most of the vegetables you will find they are generally in the case the less than that is low acid foods, okay, and they are highly prone to spoilage and pathogenic microorganism, and therefore, Clostridium botulinum is the main criteria. Then botulinum spores are heat stable and they can be inactivated only by heating to 121 degrees Celsius under pressure of 15 to 20 pound per square inch for at least 20 minutes. Okay. So, in the process consideration, what is done, like in the earlier classes we saw that is the process severity is directly proportional to the safety and the quality of the product. If you use a more severe process, then it will reduce the quality of the processed product. So, here the sterilization process takes into consideration that is two important things, that is microbiological characteristics of the product and storage requirement after the process. Okay. So, consumers are now concerned with the quality and nutritive value of the product. So, the consumer demand is a driving force for optimization of the processing conditions such as heating temperature and time to balance the safety and quality aspect. So, a heat resistant microorganism is selected and its kinetics of inactivation is determined in the product to be processed. Okay. Right. So, let us see how that is you have taken the microorganism, then what are the, let us say see that thermal destruction of microorganism, how it is destroyed. So, impact of heat treatment on microorganism is estimated from the measurement of the surviving cells against heating time. The destruction profile that is obtained allows the determination of the characteristics of the resistance of the microorganism that are the basis of the designation of the operational requirements of a thermal process. So, traditional estimations of the efficiency of preservation and disinfestation processes are based on the assumption that microbial death process follows a known evolution. So, as each species has its own particular heat tolerance, thermal operating conditions are determined experimentally and the data are processed at the number of surviving microorganisms or viable spores against the exposure time at a given temperature. Okay. So, thermal death or thermal calculation thus involves the need of the knowledge of the concentration of the microorganism to be destroyed, the acceptable concentration of microorganism that can remain, the thermal resistance of the target microorganisms, and the time temperature relationship required for destruction of the target microorganism. So, let us see microbial survivor curve. You can see here that in the graph that during heating of the food, the population of the microorganism and its spore reduces by a given factor. So, a general model for the description of the microbial curves is given as dN by dt is equal to minus k n to the power small n. So, here capital N is the number of microorganism, k is the rate constant, and small n is the order of the model. So, the microbial death curve normally follows the first order kinetics. So, n is equal to small n is equal to n. So, this kinetic model for this, that is dN by dt is equal to minus kn, or ln n by n0 is equal to kt. So, this basic model has been used to describe the survivor curve obtained when microbial populations are exposed to elevated temperatures. Okay. In many cases, however, this representation gives a linear relationship that implies that the thermal destruction phenomena is a first order reaction, particularly if you plot this log number of survivors on the logarithmic scale and the time on the linear scale. Then you will find, as you have seen in the equation 1, you will find a linear relationship. However, there are deviations from the thermal death curves are frequently observed, and the thermal death curve can also be characterized by soldiers and trails, and the deviation from the linearity can be related to the composition of the microbial population, germination of the sporulated forms, or heterogeneity of the thermal treatment. Okay. You see here again the same curve, that is line 1 is basically the curve 1 is the typical survivor curve of a homogeneous population, where there is all the microorganisms which are present in the food material, particular food, they are homogeneous, they are all similar in the term of shape, size, etc., all the characteristics, and when it is homogeneously heat treated, but which is normally not found in the case of food material. Food they contain, there is various type of microorganism, thermoresistant or thermosensitive microorganisms, spores, etc. So, the curve 2 represents that, yes, here both the thermoresistance and thermosensitive cells coexist. So, you will get for some time linear relationship, and then there will be deviation. Curve 3, it represents that, yes, the microorganism, there are certain microorganisms, they are activated by short exposure to heat, which you can see here. They are activated, so it increases and then it comes down. So, means like germination of a spore, fragmentation of chains, etc. Decimal reduction time, you can see here in the curve, that is survival curve, when CFU per ml is plotted against time, that is here in the log cycle, then you get a straight line, okay, and slope of the curve is 1 by d. When the microbial survival data are presented on semi-log coordinate, a straight line is observed, and from this you can calculate d, that is capital D, it is normally called as a d value. It is the decimal reduction time. It is defined as the time necessary for a 90%, you can see here, 90% reduction in the microbial population, or the time required for the, this is the survivor curve, to traverse 1 log cycle, or for the reduction of the population of microorganism by 1 log cycle. The kinetic model for this I have given here, log n0 minus log n is equal to T by D, or that is T by D is equal to D T by log n0 by n, or n0 by n is equal to 10 to the power small t by d. So, the first order rate constant k is inversely related to the decimal reduction time, that is small k is equal to 2.303 by d. So, by this you can calculate the decimal reduction value d value of the process. Then z value, which indicate thermal resistance of microorganism. So, if the d value, that is if you heat the commodity at various temperature, d value is at a particular temperature, okay. That is how that population decreases with time. as a particular temperature. So, you get d value at a different temperature and plot the d value versus temperature. So, this gives you the death rate curve, and here it is a slope again, and the slope gives the z value. The z value is another characteristic corresponding to the increase in the temperature that reduces a reduction in the d value by a factor of 10. Okay. The models like d is equal to d ref 10 to the power t ref minus t by z, where t, capital T, is the treatment temperature, d ref is the decimal reduction time minute at a reference temperature T ref. Okay. And there are specific d and z values for thermal inactivation and destruction or retention of enzymes, vitamins, pigments, etc. So, the reactions are components like microorganism, nutrients that has a small z value are highly temperature sensitive. Okay. And this z value is used further in the case of STST and other process parameters, etc. You can see here in this figure, generally it has been seen that the typical z values for the of 10 degrees Celsius are characteristics of the pathogenic bacteria, whereas z values of the vitamins and pigments, enzymes, etc. are in the range of 20 to 70 degrees Celsius. And this difference allows the manufacturer to determine an optimal thermal process like temperature and exposure time. Okay. And you can see here that in this line, that is the shaded line, it shows that the correspond to the STST, high temperature short time process, because in this the most of the pathogens are also inactivated. This is the pathogen detection, that is destruction line, and this is the microbial retention line, destruction line. So, here that is in this region the vitamins, enzymes, etc. are retained, but the pathogens are active. So, this is the high temperature short time. Okay. Thermal death time, from this d value or z value, etc., we can calculate the thermal death time, that is the f value. It is the total time required to accomplish a stated reduction in population of vegetable cells or spores at constant temperature, like f is equal to d log n0 by n. Okay. And from this, even 12D concept, which has been traditionally used long in the canning industry, particularly for low acid food canning in vegetables, etc., that is 12D means 12 log reduction of microorganism. For example, if in the system, in the food, there is a initially 10 to the power 12 microorganism, then after the 12D process, it will be reduced to 1. Okay, that is F is equal to 12D. So, for commercial sterility of low acid food, as I told you, 12 log reduction or 12D process are usually preferred or specified. Then lethal rate, L is the ratio of FR value, that is F value at a reference temperature T ref to F value at any temperature, log of FR by F equal to T ref minus T by Z. Okay. Or L is equal to, you can say, FR by F or T 10 to the power T minus T ref by Z. So, above equation can be used to compute the thermal death time F at any temperature when FR is known at a reference temperature TR. In most of the cases, the reference temperature, particularly for sterilization process, TR is considered to be as 121 degrees Celsius. Therefore, the FR value of any thermal process can be said as the number of minutes of heating at 121 degrees Celsius required to achieve the same thermal destruction rate ratio of a specified microorganism. Q10 value is defined as the ratio of the reaction rate constant at temperature differing by 10 degrees Celsius. It indicates how fast a reaction will occur if the temperature is raised by 10 degrees Celsius, and thus can be used to predict the expected product shelf life. For example, if a food attribute is stable for 10 weeks at 30 degrees Celsius and it has a Q10 value of 2, then it can be said that this particular attribute will be stable. Its stability at 20 degrees Celsius will be about 20 weeks. So, the effect of temperature on quality is expressed at a temperature quotient Q10 which is defined as Q10 is equal to Q2 by Q1 to the power 10 by T2 minus T1, where small q2 and q1 are the rates of quality function at two temperatures T2 and T1 respectively. So, the Q10 values have been used to describe the effect of particular quality attributes such as color, texture, flavor, etc. And commercial sterility, the term sterility signifies total destruction of all microorganisms within a medium. With commercial sterility is often used in the context of canned or specialty processed products to indicate that microorganisms related to food spoilage and public health concern have been destroyed, but there are several cases where in the product there do exist some thermophilic microorganism, thermophilic bacteria like Clostridium thermosaccharolyticum, Bacillus stearothermophilus, etc., and they have much higher resistance than the Clostridium botulinum, but they are ignored. So, in the commercial processing they are ignored, means that, but they are taken care of because they are thermal, they are thermophilic microorganisms, they require high temperature, maybe around 50-55 degrees Celsius for their growth. So, the canned food, so obviously this is taken care of, that these microorganisms which survive, their survival is not a problem, but only when they grow and multiply will create problem. So, their growth and multiplication post processing is taken care of by appropriate handling, appropriate storage of the canned food. So, by the application of heat which renders the food free of microorganism capable of reproducing of the food under normal refrigerated condition of storage and distribution, and viable microorganism including spores of public health significance, and that means the commercial sterility. Factor affecting commercial sterility, that is various rate like intrinsic factors, environmental conditions during which the food is being heated or stored post process, they all influence the microbial inactivation kinetics, etc., and most of the models they are based on the temperature levels and exposure times are relatively influenced by these factors, intrinsic as well as environmental factors. So, if you see what is the effect of heating rate, the heating rate which is generally not taken into account in the models of thermal destruction, it has been found to be a determinant parameter affecting cell survival in heating. Heat shocks are more effective than progressive heating, enabling the optimization of the thermal destruction process by increasing the thermal gradient. At a fundamental level, the effects of heating rates on membrane permeability also emphasized the fact that cell membranes are probably structurally dependent on the heating kinetics. Okay. Effect of environmental parameter like water activity and other hydrostatic pressure, etc., markedly affect the thermal treatment, okay, and its effects on the viability of the microorganisms. Microorganisms appear to be thermally stabilized by intermediate water activity value like 0.3 to 0.5, that is, if water activity is brought below this, microorganisms are completely sterilized, and the most favorable hydration condition for their destruction correspond to either fully hydrated or fully dehydrated media. As proteins are generally stabilized by extreme dehydration, it can be speculated that the thermal destruction of cells in dehydrated media is enhanced by oxidation or by membrane destabilization favored by low hydration. The major role played by membrane destabilization in the induction of the cell death can be seen in the way that passing through the membrane phase transition using a cooling step enhances cell resistance to osmotic dehydration significantly. So, the main implication of these results is that thermal stabilization of dehydrated products can be optimized, especially when heating rates are used. Now, I will briefly summarize that the quality changes during the thermal processing. Okay. Particularly, although these thermal treatments are known to have various different effects on the various quality attributes, whether physical, chemical, microbiological, nutritional, sensory, etc. But I will just take up briefly the nutritional, its effect on nutritional and sensory quality attributes. Like in the nutritional quality attributes, its effects on protein, if you leave, obviously protein denaturation, hydrogen bond rupturing, conformation changes in the protein, there may be oxidation reduction with reducing sugar and degree of protein denaturation depend on the level of heat treatment, and total crude protein content however is generally not affected by canning. But there may be conformational change and other thing which may influence the other functionality of the proteins, etc. Lipids, normal heat processing has generally no effect on the fat content. Hydrolysis reactions can occur, but has no adverse effect on nutritional value. Unsaturated lipids are more prone to oxidation than saturated one under heating in presence of water. The carbohydrate level of total and available carbohydrate are largely unaffected during thermal processing of fruits and vegetables. In general, their effect are more related to how these carbohydrates are interacting during processing with other food constituents and to the overall eating quality of the food stuff, how it is influenced. Starch gelatinization or increased digestibility, etc. of the starch are some of the changes that may take place in the carbohydrate during thermal processing. As far as the effect of thermal processing on minerals is concerned, normally minerals are susceptible to changes in bioavailability due to interactions with other food components. Major changes that can occur in the mineral levels on canning are caused by movement between the food stuff and the canning liquor. Effect on vitamin, can we see that most of the vitamins are unstable under conditions of heat and are susceptible to losses during the canning process. The fat soluble vitamins are generally more stable than the water soluble vitamins, but the losses can occur during canning due to oxidation, also losses of vitamins, they may get oxidized. Now, the effect on sensory attributes, like more important effect on the flavor, if you see that there may be lipid oxidation, that is unsaturated fats are degraded under the conditions of oxygen and heat, leading to both desirable and undesirable flavor. There may be Maillard reaction, that is it depends on the temperature, pH and the water content of the commodity. Taints, there may be various off flavor development due to contamination from the environment, etc. Texture of the material may be influenced, that is how even starch gelatinization, swelling of the starch granules when amylose and amylopectin gives a firm gel, and translucent paste products after cooling, which may be in the peas, beans, etc. starch or even potato, that this texture changes when you boil a potato, you see how this texture gets changed. Similarly, pectin changes, loss of semi-permeability of the cell membrane, solubilization and breakdown of pectic substances in the cell wall and middle lamellae. It could lead to improve palatability of the food, or over processing can cause excessive softening of the fruits and vegetables. Similarly, color like chlorophyll, there may be breakdown of natural pigments which leads to, in the particularly in the case of vegetables, etc. there may be browning or other associated changes due to the structural change in the chlorophyll. That is, the magnesium from the chlorophyll oil is removed, which gives the phytins, etc. and this causes the green color to change in the brown color or dark brown color. Carotenoids, that is there, it may be oxidized, that oxidation and isomerization under the conditions of heat and low pH, that is, this may also influence the changes in the color. Okay. So, now only I will summarize this lecture, that as blanching and pasteurization are the mild thermal processes, where canning, baking, roasting and frying are the most severe thermal process. Hot water blanching or steam blanching is generally used for enzyme inactivation, whereas pasteurization, sterilization and aseptic processing are used for microbial inactivation. Thermal process calculations for low acid food is required and is mostly based on the inactivation of Clostridium botulinum, thermal dead time, D value, Z value, F value, Q10 value and lethal rates are the concepts that need to be properly understood. during thermal process calculation, and thermal process mostly affect the nutritional and sensory properties of the fruits and vegetables. So, it is very, very important that the severity of the thermal process should be properly controlled, that is the thermal process conditions, they are properly calculated and then appropriately, that is the heated, that is appropriately managed during processing, so that the undesirable factors like enzymes, undesirable microorganisms are inactivated, but at the same time quality factors are retained to the maximum value, maximum level. So, these are the references which are used this. Thank you all very much.